Good morning and warm welcome to all the participants. It's our pleasure to have you with us. Today, the topic of deliberation is pesticide contamination in aquatic ecosystem, a concern merely considered. And we have with us Dr. Sheikh Imtiaz. Before he could begin with the presentation, I would like to give you a formal introduction of our esteemed guest. Dr. Imtiaz Qayyum is working as assistant professor in the division of AEM FOFY's Coast Kashmir. He's working as a toxicologist and he has worked on various aspects of environmental toxicology, monitoring assessment in vitro toxicological studies and behavioral toxicology in fishes. Being holder of prestigious Inspire Fellowship, he has also received Best Young Scientist National Award from prestigious IRDP group of journals, Chennai. He has authored four books related to different aspects of fisheries science, and practical laboratory manual for aquatic toxicology. He is working on different aspects of environmental toxicology through externally funded projects and has also published a number of research papers of national and international repute. Being engaged in research, teaching, and extension activities, he is holding various administrative assignments given by his respective institutions across Kashmir. Sir, you are audible as well as visible, and your presentation is also visible. Please start, sir.
Thank you, dear Majid sir. Good morning and very warm welcome to all of you. My topic today is the pesticide contamination, which is actually one of the emerging topics all over the world as far as our lakes, our ponds, our rivers, our oceans and seas, and other water bodies are concerned. Actually, these contaminations caused by pesticides and other xenobiotic compounds, they are merely addressed, they are seldom noticed, and which are not actually apparently visible in our water bodies. When a limnologist or aquatic ecologist has a look on any water body and sees the apparent change in the water body as far as its odor is concerned, its smell is concerned, its taste is concerned, uh, or its color is concerned, immediately a limnologist gets an idea that this water body might be suffering from increase in nitrates and phosphate levels, which will cause pollution to it. But contamination, by contamination, we mean those xenobiotic compounds which are actually not visible, but when they are present, and even if they are present in very small quantities, they are present in very small residues and in any compartment of the aquatic ecosystem, they pose threat to the aquatic flora and fauna. And not only that, but they are actually very, very harmful for the humans, which are at the top of the food chain, consuming all these commercially important plants and animals. For example, you have a pesticide which is present in any column of the lake or water body. It is getting bioavailable, it's getting exposed and ready to be taken in by the commercially and economically important plants and animals like fish, like you have water chestnut, like you have nilumbo, like you have edible macrophytes. And those plants and animals, when they are being eaten by humans or they are eaten by higher organisms placed at the higher trophic levels, uh, which are in turn eaten by human beings, they get transferred, not only bioaccumulated by but biomagnified as well. Therefore, even if present in small quantities, pesticides pose threat to our natural ecosystems and threat to our aquatic plants and animals. In this presentation, we will be briefly uh, clearing the concepts regarding pesticide contamination, which is a concern actually, but that is merely considered, because since presence of pesticides in aquatic ecosystems is not visible, therefore we seldom notice those contamination and avoid uh, actually dealing with uh, those research areas around the world. Although now this is an emerging subject, people are actually getting involved in it, and we are getting a lot of data these days in and around the country regarding contamination levels and pesticide residue levels in our natural ecosystems. Let us quickly move to the first slide. And first of all, let me get the basic information about the pesticides. And this pesticide is actually derived from a Latin word called sida, which means to kill. Means actually these pesticides, they are used to kill those unwanted organisms or insect pests which are actually harming our crop. Okay, uh, we use those pesticides, we use those insecticides, we use those rodenticides and weedicides to kill the unwanted organisms, be it of plant origin or be it of the animal origin in the crops. But unfortunately, those pesticides, those chemicals, they are carried away and they are washed away into the aquatic ecosystems. Uh, Actually, there are various factors which are responsible for movement of these pesticides into aquatic ecosystems. One of the important and foremost factor is that the farmers, which actually spray these pesticides to their crop, they don't use these pesticides judiciously. By judicious, I mean they don't use these pesticides in accordance with the spray schedule, in accordance with the scientific guidance that is being prescribed for a particular pesticide. For example, if one pesticide A has a recommended dose to an apple crop or to a coconut crop or to rice fields, pesticide A has recommended dose of that it should be sprayed on the crop at the rate of 2 ppm, means 2 milligrams per liter. Fine. But farmers being ignorant, most of them, and being illiterate, most of them, they randomly mix this pesticide without the actual this measurement of this pesticide in accordance with the spray schedules prescribed by the scientists or agriculturists, which results in the accumulation of these pesticide residues in the crop, in the soil, and they are being carried away and washed away uh, to aquatic ecosystems by rain or by drifts or by leaching or percolation. Uh, these pesticides, they have a mechanism of action, particular mechanism of action to kill any insect pest, but the same pesticide, when it is reached in the aquatic ecosystems, water bodies, then it shows the same mechanism of action to the non-target organisms like fishes, like macrozoobenthos, like zooplanktons and phytoplanktons, thereby cause either a rest of their life or uh, physiological dysfunctions in them by acting through the same mechanism of action. Quickly, we will say the pesticide is defined as a chemical agent that is used to control or destroy the insect pests. I got one of the important definitions, which is very much actually uh, a definition which encompasses all the aspects of pesticides, is that pesticides are substances used by human beings to kill or deter organisms or pests that threaten our health and well-being, the health and well-being of pests and livestock, and cause damage to crops. Now, these crops which were being sprayed by the pesticides, non-judicious use of pesticides, indiscriminate spray of the pesticides, those pesticides which were recommended for one particular season, they were supplemented with other pesticides as well, of the own knowledge of the farmer, without the consultation of scientific staff, without the consultation of agriculturist or plant pathologist, these pesticides get washed away into aquatic ecosystems, because aquatic ecosystems, they are naturally uh, uh, placed at the depression as compared to the land. Similarly, they become ultimate sinks, where all the dirt, where all the contaminated this materials, where all the sewage, and where all the nitrates, phosphates, they are being washed away and dumped into these lakes when their rainfall occurs, or aerial drift occurs, or land drift occurs, or leaching or percolation occurs. Most importantly, when an orchard or a vegetable garden or a cropland is placed near the watershed area of any water body, and which is being indiscriminately sprayed by these chemicals, those chemicals readily wash away and get percolated down to the waters. Uh, actually, uh, let us quickly move to the classification of pesticides. They are being classified on the basis of their mode of action and on the basis of the target organisms. You perhaps all might be knowing that those chemicals which act on rodents, those rodents which kill our crop, which destroy our croplands and plants, they are called rodent sites. Similarly, for unwanted weeds, we spray weedicides. For insect pests, we spray insecticides. Acaricides are actually those pesticides which are sprayed on ticks and mites that are harmful for our crop. And fungicide, you all know, they act against the fungus. These are the examples there. You can quickly go through them as well. On the basis of mode of action, we know we have organochlorines. Actually, they are the persistent compounds which are known to deter the environmental life, and they are more persistent in the environmental compartments. They remain for longer times in any of the environmental component, be it soil, be it water, be it sediment, be it fish or macrophyte, thereby cause a lot of physiological dysfunctions if present in small quantities. And if they are present in higher quantities, they can lead to mass kill or a mass mortality of the crop or fishes as well. They act on the neurons by blocking the potassium channels, the nerve impulses, thereby causing paralysis in the organisms. Now, see, those insect pests, for example, organochlorines, they were meant to cause the neurological dysfunctions, arrest of sodium potassium pumps in the insect pests. But when they are washed away into the aquatic ecosystems, they act by the same mechanism uh, to cause mortality of fishes as well, mortality of other organisms as well. Similarly, organophosphate compounds were known that they cause inhibition of acetylcholinesterase, an enzyme in the synaptic clips of the neurons, which results into the accumulation of acetylcholine and arrests the normal uh, transmission of nerve impulses. Now, these organophosphates, when they act on the fishes, they again cause the inhibition of ACHE in fishes when these pesticides reach the aquatic ecosystems. Carbamates similarly have same mechanism of action like organophosphates, but they have a difference from organophosphates in the sense that they are less persistent in the environment. They don't accumulate in the environmental compartments and are readily degraded into the non-toxic forms as compared to organophosphates. And pyrethroids, they are the derivatives of natural pyrethrin plants, and they also act on the nervous system of the insect pests, thereby causing arrest in their neurological functions. Actually, there is a sequence in this organochlorine and organophosphates in carbamate and pyrethroids, in the sense that these chemicals were invented and discovered actually on account of their persistence. In the earlier 19th century, or sorry, late 19th century or early 20th century, organochlorines were used as warfare agents to cause arrest of neurological functions of the armies around the world, which were indulged in world wars and all. Similarly, when we discovered insecticidal properties in them, we used them on our crop, but simultaneously we came to know that they are very much persistent in the nature, thereby cause environmental degradation, environmental contamination, deterioration of any component of the environment in which they prevail. And countering that, scientists discovered and invented there's some chemicals which were less persistent than organochlorines in the form of organophosphates. Similarly, organophosphates were less persistent than organochlorines, but they had a complex there's physiological problems associated with them in non-target species. For example, although organophosphate compounds like dimethiate and chlorpyrifos, they are present less persistent as compared to organochlorines like DDT and endosulfan. Uh, but they have a high toxicity to the mammals and to other organisms which are present in the natural ecosystems. Therefore, on account of their high toxicity, they were replaced by carbamates. Not fully replaced, actually, uh, but they were supplemented by carbamate pesticides, which showed less persistence to the non-toxic organisms. Similarly, pyrethroids are the natural derivatives of pyrethrins. They are least persistent of all the all the insecticides which are mentioned here. Uh, Now, uh, the entry of these pesticides into aquatic ecosystems are actually from two important sources called as point sources and non-point sources. Point sources, all know that those sources which are actually notified and specified. For example, we have a sewage treatment plant from which treated sewage is allowed to enter into an aquatic ecosystem. Now, this is known actually source from which possibility of entering of pesticides or a metal or any other chemical is can be possible. Similarly, we have here, you just see, this is the example of our Kashmir. Actually, you have a famous Dal Lake and houseboat is here. Fine. And these are the vegetable sellers, the floating vegetable sellers. These vegetables are taken up from these gardens. They are called as the floating gardens. Uh, these gardens are present in the lake, within the lake, where a lot of pesticide is being sprayed. And you can see the apparent deterioration of the water quality here uh, in the back of these boats. And when these pesticides are sprayed non-judiciously, indiscriminately, without consideration of any scientific guidance, they will leach down, they will percolate down, they will reach our aquatic ecosystems, and they cause environmental contamination. But since this is not a specified area where we can locate actually where from pesticide is entering the aquatic ecosystem, this is categorized as non-point source of pollution. Similarly, we have a hotel here, fine. Uh, Sewage coming out of this hotel and kitchen waste coming out of this hotel, which is actually a known place where from this sewage and all outgoings are dumped into the lake. They are the point sources of pollution. Now, here we have an apple orchard where obviously a lot of pesticides are being sprayed. They will reach the aquatic ecosystems, but we are not specified actually where from they will reach our aquatic ecosystem. Therefore, this is a non-point source of pollution. Point sources are uh, uh, include wastewater treatment facilities, runoff during non-overflow conditions, combined sewer overflows during wet weather, manufacturing facilities, and spills, and leaks on farms or other areas where they are stored and handled in bulk quantities. And non-point means they are less specially localized. We are not able to localize them. We are not able to point out them where from actually they come and they reach our aquatic ecosystems. Point sources are defined, but non-point sources are not defined actually. For example, in developed countries like America, you have end-to-end crop fields. You have end-to-end your agricultural lands, and those are being sprayed by are being sprayed by pesticides through aerial mode. Where you have small there's helicopters which spray the large amount of pesticides on those agricultural crops. But those pesticide droplets remain suspended in air as well on account of their bondage with the particulate matter or cloud or water droplets. They remain suspended. When a strong wind comes or a strong rain flows that area, they are washed away and taken away to the water bodies as well. Now, you don't have any defined point where from you can specify that these pesticides are entering into the aquatic ecosystems. Therefore, they will be regarded as non-point sources of insecticide or pesticide contamination. Now, coming to various factors that are responsible for bioavailability of a chemical contaminant in our aquatic ecosystem. Means if a pesticide or an insecticide that enters our aquatic ecosystem, what will happen to it actually, and where will it go, and what harm it is supposed to cause to our flora and fauna in the aquatic ecosystem. Those factors which are governing the bioavailability of these pesticides, they actually include number one, the chemical speciation. The chemical speciation of any pesticide is actually one of the important governing factors which will define whether the pesticide is harmful for aquatic ecosystem, whether the pesticide is harmful for important plants and animals, and whether this pesticide will pose threat to humans later on as well. Chemical speciation means the chemical bondage and the elements which are actually present in the uh, in that molecule in that pesticide. Let me mm, go to this slide. I will show you this. These are some of the pesticides actually. Look at it. Let me come to this. This is DDT. All might be knowing DDT. All might be knowing endosulfan. All might be knowing eldrin and dichlorobos. These are one of the important and commonly uh, named pesticides or insecticides which have been used by the people all over the world and from a lot of time actually. If you will closely look into the chemical structure of this DDT, for example, let me take example of this DDT. This is an aromatic carbon having two benzene rings, and associated with it are some five chlorine atoms. You just take the sigma rule, you just take a right hand thumb rule that the more number of chlorine atoms present in a molecule of an insecticide, the more persistence it will show in the environmental compartments. Means by persistence, I mean it will have a greater half-life. And you have one of the important uh, tool by which you can measure the persistence or the lipophilicity or hydrophobicity of any uh, insecticide, and that uh, is a formula which is called as Octanol, water partitioning coefficient, and is specified by log of KOW, and it is amount of contaminant retained in an octanol, normal chain octanol, over the amount of contaminant in the water. I will give you this simplified example of it. You have a beaker, and in which you uh, drop water, normal water, and you drop normal chained octanol. Since these two are immiscible, they will not get mixed with each other. They will form a layer in between this layer. Is my cursor moving around? Is everybody able to see? Sir? Am I audible to everybody?
Yes, sir, you are audible.
Okay,
Yes, sir. Yes,
okay,
sir. Continue. Continue, sir.
okay, okay, fine. Now, you have two immiscible liquids in a beaker, and you will drop some insecticide like this DDT. This DDT you will drop in this beaker. Fine. You will have to see in what compartment this DDT will go. This is very important actually. You just all need to know this in order to comprehend what actually happens to pesticide when it enters the aquatic ecosystem. You have an octanol, I will repeat. Then you have water. There are two immiscible liquids which will not mix up with each other. And you drop any chemical, any insecticide, any pesticide here in this beaker. And you will see whether that insecticide will go to this compartment of an octanol or it will go to the water. If it suppose goes to an octanol and it remains suspended in the upper column, means it is lipophilic, means it is fat loving, because an octanol is an organic uh, compound and that insecticide was uh, organic loving, fat loving, lipophilic. Therefore, it got settled down in this column. Similarly, if it will be hydrophilic, means water loving, it will settle down here in the water column. Fine. We calculate this actually. The chemicals which have a value of log of KOW more than 5 or 5, they are lipophilic actually. And which have below the 5, this value of log of KOW, they are hydrophilic. Now, keeping this thing in view, just imagine if a pesticide or insecticide which is lipophilic, which is fat loving, when it reaches the aquatic ecosystem, it enters into a lake. Where it will go? It will go to number one, macrophytes, phytoplanktons, zooplanktons, fishes, because they are all organic and they have more or less some fat available there in their bodies. It will tend to enter into the fatty compartments of these organisms. Moreover, we have one more thing, uh, that on floor bed of the lakes, on the ground of the lakes, you have lot of microorganismic population over there, which is called as microbial consortia. Okay, and those microbial consortia, they excrete some organic substances around them that are called as extracellular polymeric substances. Look at this. This is the microbial consortia, and this is the organic layer, which actually is secreted by these microorganisms. This is the scanning electron microscopic photograph of these biofilms or EPS, extracellular polymeric substances. These are also organic. So when insecticide enters into a lake, it will tend to get absorbed to this extra polymeric substances as well. So keep in mind, number one, it will get settled on the body of microphytes, zooplanktons, phytoplanktons, fishes, uh, emergent or submergent or rooted or floating, free floating weeds, or aquatic, no, this extracellular polymeric substances as well. This is when an insecticide will be lipophilic. It will be fat loving. It will get absorbed or suspended therein. In either case, fishes will try to grab these extracellular polymeric substances. You know, we have a lot of fishes. For example, we have Sirenus mrigala, that is a bottom feeder. We have Cyprinus scarpio communis. We have Cyprinus scarpio specularis and nudus. These are all bottom dwelling fishes. And as a part of their food, they scrap the floor bed of the lakes because it has a lot of humus present, organic material present in it. Or some fishes which are not even bottom dwellers, they, as a part of their accidental food or emergency food in case of drought conditions or all, they try to eat and gallop these sediments. And if you have contaminated water bodies, uh, these pesticides will enter into the fish body. Similarly, if the pesticides are adhered to the macrophytes or zooplanktons or phytoplanktons, these pesticides, these zooplanktons or phytoplanktons, they are being eaten by the fishes or eaten by other organisms, which are in turn eaten by the fishes in the food chain, and they enter the fish body in any way. And we know that we eat a lot of fishes. Humans are fond of fishes, and those insecticides directly get transferred to the human population. This is the condition when the pesticide will be lipophilic. If the pesticide is hydrophilic but water loving, it will not get settled to any of the column, any of the compartment mentioned earlier, but it will remain suspended in the water. Still, when it will be suspended in the water, it will be directly taken up by fishes through gills, or it will be exposed orally, or it will be exposed to the fishes dermally. And in either case, whether the pesticide is lipophilic or hydrophilic, it will be taken up by the fish population or important these plants or macrophytes, and which are in turn being utilized by human beings residing around the lakes. And these pesticides get transferred, bioaccumulated, and biomagnified in the human population as well. So, number one factor which is responsible for a pesticide to be bioavailable for any compartment in the lakes or water bodies is its chemical nature. Take it as a sigma rule here. More chlorines, more it will be lipophilic. That's why organophosphates, organochlorines, that is, they were called as organochlorines because they were having more of the chlorine in their benzene rings attached. Therefore, they were more persistent in the nature. They were replaced by organophosphates. And there are various organophosphates as well, which have chlorine atoms in their benzene rings. When chlorine atoms will get increased in any of the compounds, it will tend to show higher persistence in the environment, and it will tend to show higher half-lives in the environment as well. There are some examples that DDT that I mentioned is its half-life is 150 years. For example, if you have DDT present at the rate of 2 ppm per liter of water in a lake, means 2 ppm means in 1 liter you have 2 milligrams of DDT. And you imagine how many liters of water will be in a lake. There'll be innumerable number of liters, several thousand liters, we don't know. And in every liter you have 2 milligrams of DDT present. Okay, and this every liter of water is posing a threat to the fish when fishes are being exposed to this water. Okay, and this 2 ppm will get reduced to 1 ppm, means its half-life after 150 years. Imagine, means after 150 years still you will have exposure of 2 ppm of this DDT to fishes. You can yourself imagine how much uh, disastrous it could be for the fish population. Even if it is present in very small amounts, it is still getting exposed to the fish population for 150 years. And even if not causing mortality, mass mortality of fishes, still it will lead to endocrine disruption, neurotoxicity, and reproductive failure, and various other physiological dysfunctions in there. Similarly, you have chlorpyrifos of 30 days, dimethylate of 8 days, aldicarb 5 to 10 days. There are several examples I have mentioned here. After going through the literature, you can yourself see that even if they are present in very small quantities in our lakes, they are exposed to our fishes, to our economically important, commercially important macrophytes, aquatic foods, other than the flesh and meat. And they are very much disastrous to them as well. This is the pictorial representation of half-life. You all might be knowing it. I need not to go into detail into this slide. Now, there are certain factors like photolysis or oxidative reduction, oxidation or reduction of chemicals that occur on the surface of the water body. That's why when we carry out the residue analysis of these pesticides in the water bodies, we don't actually find a lot of pesticides present on the surface because they get destroyed by the UV radiations and they get destroyed by the sunlight and oxidation and reduction uh, processes. But when a pesticide enters into the water body, it quickly moves into the column and bottom of the lake. Right? That portion or that magnitude which is remained upwards on the surface, that gets destroyed actually. And sunlight also plays a governing factor regarding the bioavailability of any compound in the aquatic ecosystem. Extracellular polymeric substances, I gave a brief introduction about it. They are the natural polymers of high molecular weight secreted by microorganisms to live in a colonial form. Because we know that they live in consortia, they live in colonies, therefore they need to secrete some organic substances around them, which will not act as a media for them to be in a cozy state, but for the transport of chemicals and waste materials in and out as well. Therefore, these being organic, they quickly capture those insecticides or pesticides which have log of KOW, means octanol water partitioning coefficient more than 5, or they are lipophilic. Now, port of entry of pesticides in fishes. I told you in any case, if it is lipophilic or it's hydrophilic, both will try to harm our fish organisms. In fishes, we have three portal of entries, three routes by which through which pesticide can enter. One is dermal exposure, means if a pesticide is hydrophilic, it's water loving, it will remain suspended in the water column, it will get constantly exposed, and it will get constantly in touch with the skin of the fishes, and it will cause uh, lesions, and it will cause disruption of the lipid bilayer uh, of the cell membranes. You might be knowing that we have different kinds of fishes. One fish having scales like common carp, and other have small scales like we have eels, or we have trouts, or we have shadowthracine fishes. Quite obvious to say that those fishes having small scales or having feeble outer layer of their skin, pesticides will enter their bodies more. That's why we see on and off in the normal indigenous fishes like chilothracines of Kashmir and rainbow trouts. If they are exposed to pesticides, they develop strong skin lesions as compared to common carps. Those common carps which are young in age, they are juvenile, like in fry or fingerling stage, they have feeble scales. On them, pesticides also enter very quickly and cause disruption of their skins. Similarly, fishes take up water, uh, and that water has to be passed through gills, and gills are very sensitive in transfer of chemicals and all to the body. These pesticides enter to the gills as well, and from gills they will enter into the whole body of the fish. Similarly, if pesticide contaminated feed is being supplied to fishes, uh, they absorb that pesticide through GIT and cause various physiological dysfunctions. Now, there are various effects of pesticides. We know they will cause disruption in the histological architecture, means histopathological alterations. They develop aberrations in the chromosomes and development of uh, the genotoxicity. We have a very cheap and effective indicator of genotoxicity called as micronucleus, uh, which is developed in the fish hematocytes as a part of indicator of genotoxicity in fishes. It has been seen, we have been working on the genotoxic aspects of pesticides in fishes, that they cause large amount of um, breakage in the DNA in the fish hematocytes that results in the development of genotoxicity in fishes. Similarly, it will cause anemia in fishes. It will cause lower values of hematocrit and this MCH and MCHC values, thereby causing hematological alterations as well. Similarly, they will try to increase the values of ESR, uh, and means higher values of ESR is indicative of insecticide toxicity in fishes. Similarly, WBC count means is an indicator of the pathological conditions, and we have seen that a lot of WBCs like monocytes and all, they get increased from the normal when fishes are stressed with the pesticide toxicity. And you have biochemical alterations and larvivorous ability alterations, and you have hypoxic conditions, neurotoxicity, as I told you earlier, in case of organophosphate and carbamate insecticide exposure, and you have endocrine disruption, which is caused by fishes. And we are working on behavioral aspects actually. Behavioral toxicology is very important and primary tool by which we can apparently see change in the behavior of fishes when they are exposed to pesticide contaminated waters. Therefore, these are small briefings about the various uh, deleterious effects, various physiological dysfunctions that are carried by pesticides in fishes. We cannot go into the detail, that is out of the scope of this time, because we might be running short of time. And actually, they are the area of the research, and everybody can take one aspect among these and work with in-depth information and insight full research to get various aspects out which are not known yet to the fisheries toxicologists. Uh, these were the effects of pesticides on fishes. Okay, if they are present in small quantities, they will lead to physiological dysfunctions. But um, human aspects where we are concerned of is actually the biomagnification and bioaccumulation of these pesticides in the food webs or food chains, which actually lead to uh, the various physiological dysfunctions in humans as well. Bioaccumulation means uh, trophic level, in one trophic level, how much of pesticides is retained in by the organisms of that trophic level. And biomagnification means when this pesticide is transferred, this insecticide is transferred to the next trophic level, it not only gets accumulated in them, but it also gets magnified in them. For example, you might be knowing in ecology the pyramids of numbers. Pyramids of numbers means you have mm, a lot of individuals present at the base of that pyramid. Means uh, in herbivores you have a lot of numbers, and those primary carnivores which are placed at the top of the herbivorous level, they are comparatively small. Similarly, when you go on increasing this food chain, or you are going to climb from one trophic level to another, the number of organisms gets reduced. It does not mean it only gets reduced. Actually, those pesticides which are accumulated at the base of the that pyramid, it also all gets transferred into the higher trophic level, and it gets concentrated and magnified there. Similarly, when humans are placed at the top of the food chain, all the contaminants which were present at the base of the food chain in fishes or in aquatic macrophytes, that reaches the human population in not only bioaccumulated form but also in biomagnified form as well. And these are very in-depth concepts which need to be understood, that even if present in the small residues, small quantities in water bodies, they'll tell upon the human lives as well. We say that those populations, those hamlets which reside in and around the lakes and that are contaminated with pesticides, we see a lot of incidences of cancer among the residents of those areas, because they frequently take fish, they frequently take the aquatic plants and all for their consumption, which readily gets accumulated in them, the contaminated food, through the contaminated food, which cause cancer in them. Therefore, complete monitoring and assessment of our water bodies is imperative, and regular monitoring and assessment. We have to chalk out the plan how to regularly monitor the level of pesticide residues in lakes. Therefore, we will be making and chalking out a plan how to decontaminate our lakes. There are various methods by which we can get rid of these residues, rid of these contaminations, uh, which are actually not in the scope of this lecture. That might be dealt later on. But the thing is, we need to have a complete environmental monitoring assessment by which we will be able to see how much lake is contaminated and how much this contamination is transferred to the human populations in and around the lakes, which will cause serious problems, health problems in them. In fishes, we know that it tells upon the survival and growth. It will alter their behavior. It will alter their this growth. It will restrict their fecundity, and it will arrest their normal fetal growth, that is called as teratogenesis. It will cause deformity in their uh, this skeletal system, and this is actually the one of the repercussions of the pesticide intoxication in fishes. And in biochemical parameters, we have oxidative stress like cortisol and interleukins and toll-like receptors. They are being disrupted by the fishes. Similarly, the normal protein values and liver enzymes, uh, mostly like lactate dehydrogenase, like bilirubin, and like aspartate aminotransferase, these uh, uh, enzymes show alterations from the normal when fishes are exposed to the pesticides. Therefore, pesticide exposure not only restricts the growth, not only restricts the survival, but also hampers the normal crop and tells upon the human lives as well. Actually, this is a serious concern which I earlier mentioned in my topic, merely considered, and we don't even take our thoughts, take our considerations to this part of the research. Bioaccumulation, I have told you, bioconcentration is actually the bioaccumulation, but this occurs through the medium of water. And bioconcentration factors, we see the amount of contaminant which is retained in fish tissues after it was being taken up from the contaminated water. We in fisheries deal with bioconcentration factor more rather than bioaccumulation factor. Now, coming towards my last slide, this is actually the recommendations. And recommendation is, number one, we should be using pesticides judiciously in and around the lakes. What do you mean by judiciously? It is actually with the consultation of the scientific staff, agriculturists, and those who are dealing with uh, formulation of spray schedules and who are dealing with actually the formulation of recommended doses of pesticides. Farmers should be made aware of the things that they uh, randomly use uh, pesticides. For example, I visited somewhere in Kashmir someday. We had a survey in and around lakes and how farmers use pesticides. I asked one farmer, and how much of the pesticide you are using? A particular pesticide was being used by him. He told me, I just put 4 to 5 this spoons of this pesticide into one bucket of water, and actually that was 5 to 6 times more than the recommended dose which was prescribed by the agricultural university here. Okay, therefore, in ignorance, in illiteracy, and without any practical this and scientific guidance, they indiscriminately use these pesticides, which actually tells upon our aquatic ecosystems and our lakes and our water bodies. Therefore, farmers should be made aware of judicious use of pesticide. We should restrict our cells for using indiscriminate pesticides. We should also try to locate point and non-point sources. Actually, the non-point sources are diffused. For example, you have an orchard uh, in the catchment area of lakes. That orchard allows entry of pesticides to the aquatic ecosystem through percolation. So leaching. Therefore, we should develop techniques how to restrict the entry of those pesticides in the lakes. And point source is really specific, defined. We should stop the entry of pesticides through the point sources to our lakes. Now, this is third point is very important. Switching over to organic farming. Organic farming means we should restrict or completely abandon use of pesticides. But practically speaking, absolute organic farming is not possible. Uh, Therefore, we need to spray some pesticides. What can have alternative if a pest is malacing our crop and is actually destroying our crop? We can restrict the use of fertilizers. We can restrict use of uh, NPK, and we can supplement those NPKs through manure and all that through vermicompost is a very good technique. But uh, switching completely to organic farming is actually practically impossible. Therefore, pesticides will be there, but those pesticides need to be sprayed uh, with the recommended schedules and with the consultation of scientific staff. And last but not the least, that we should specify which pesticide or insecticide is least persistent in our environmental compartments. Those environmental compartments which are actually related to aquatic ecosystems. And when we come to know with the consultation of an expert that this insecticide or pesticide is least persistent in an environment, we can use this insecticide, but with its limited use in the watershed area. So this by this I conclude my presentation, and I'm open to questions. You can ask questions whosoever wants to.
There's a question in chat, and I think Dr. Kamlesh Yadav has raised his hand. He can ask question via voice as well. Just let me open the voice so that, yeah, I think they can directly ask question on chat as well as voice. If you have any questions, please ask.
Here's a tip. Use
Yeah, please ask. There are questions in the chat. Sir, what is, yeah, you could see.
Shall I answer first in the chat box?
Yeah, that would be fine. All the while people can ask on microphone as well.
Mr. Maya Raman.
Yeah.
Uh, uh, Actually, warfarin is this is a rodenticide. By rodenticide, we means that we have a lot of rats and rat-like species that destroy our crops. Okay, and that cause heavy mortality of our crops. And they not only do that, but they only cut the trees, crops, plants as well. So we spray warfarin. Warfarin is a rodenticide. It acts on the liver. Actually, it is metabolized by the liver. In liver, it is metabolized by cytochrome um, 1A1, cytochrome 1A2, and cytochrome 3A4. These are the cytochromes present on the mitochondria of any cell. These are being metabolized by the livers. But this warfarin, they can be harmful to our uh, fishes as well when it reaches the aquatic ecosystems. Although the octanol water partitioning coefficient, the log of KOW, is only 2.70, means it is hydrophilic, it is water loving. Okay, and its half-life is 8 to 24 hours in the soils. But if it reaches in our aquatic ecosystem, since it is water loving, I just told you, it will remain suspended in the water column. Okay, and while being suspended in the water column, it can be directly taken up by the fishes. It can be directly taken up by our aquatic organisms, and therein it will cause the same destruction that it was supposed to be caused to the rodents in the watershed area. Fine. Uh, Therefore, it's very harmful, and it gets metabolized in the liver, and thereby it causes the bioaccumulation in the livers as well. Hope this is the answer to the question. Dr. Devendra Harkal, how to track oh, it's getting- how to track bioaccumulation and biomagnification, source level of pesticides. Dr. Devendra, actually, when we carry out residue analysis of pesticides in the lakes, how we carry it, we will take samples from the sediment. I'm working on a project this time that is being funded by DST. We're actually trying to develop the baseline studies, how much of a pesticide or pesticides they are present in the sediments. Okay, sediments. Now we will track a sediment sample for pesticide, and we will get, suppose, the levels of pesticide in the sediment. Then we will track the water which is collected from the same place. Okay, and we will analyze it through the chromatographic techniques. For example, we will go for gas chromatography, or we will go for HPLC, high performance liquid chromatography, and we will see the amount of pesticide present in the water. Then we have the bio concentration factor, that the formula which I already mentioned in my presentation. The amount of pesticide that was in the sediment and amount of pesticide that was transferred from sediment to the water. Actually, it will give us the amount of bio accumulation or bio concentration from sediment to water. Okay. Now, we will track the fishes. We will analyze those fishes for residue analysis. Then we will see how much of amount of insecticide or pesticide is present in the fishes. And we will envisage that from water, it got transferred to the fishes. Therefore, we can check the amount bioconcentration factors from sediment to water and water to fish. And if you want to see the bioaccumulation and biomagnification in the humans, therefore, you need to carry out the residue analysis of blood samples of human beings, which are frequently using contaminated waters or contaminated fish. For example, in the developing countries like India, we have a lot of hamlets, a lot of people in the hamlets, they use water from the lakes for their potable purposes, for drinking, etc. Okay. Now, if you carry out the residue analysis from the blood samples, from their fatty tissues like cheeks, like tummy, like buttocks, like thighs, because if pesticides are lipophilic, they will get accumulated in the fatty layers of the fishes. We will take the samples from them in humans, and we will take the samples from the water. Then we can see the bioaccumulation from water to the human beings. Or if they take contaminated fish, we can see bioaccumulation from fishes to the human beings. Hope the answer to the question. Siva Sankar, P. How to diagnose immunoendocrine disruption in fish caused by pesticides? Yes, there are actually methods. This is endocrine disruption is, for example, if you supposed to see the mechanism of action of organophosphates and organophosphates and carbamate pesticides. I told you that they have a property of uh, inhibition of acetylcholinesterase enzymes in the synaptic clefts, okay, which cause accumulation of acetylcholine. Those acetylcholine was supposed to be hydrolyzed by acetylcholine esterase, and after hydrolysis, it will allow the transmission of nerve impulse from one neuron to the another. But since acetylcholine esterase was inhibited, therefore it did not allow the hydrolysis of acetylcholine, which caused accumulation of acetylcholine in the synaptic clefts, that causes paralysis in the fishes. Now, there are several kits. It actually comes under the biochemical assays. You will use biochemical analysis, and you can use the level of acetylcholinesterase present in liver, in brain, in kidneys, uh, in gills, etc., by which we can diagnose the level of immune disruption. Okay. Similarly, you have kits for endocrine disruption as well. Hope the answer to the question. Chris, actually, chrysolite Bagthasing. Actually, I'm sorry if I'm not pronouncing this name correctly. What is the level of pesticide detected in fishes of Kashmir? Yes, actually, this is a very important question. Actually, sir, I am aquatic toxicologist. I was recently I was recently appointed some 3 years back, and I had my PhD done. I was the first person to report some pesticides in the uh, water bodies. You might be knowing famous Dal Lake of Kashmir. We have a good publications for that study as well, published in good journals. Those pesticides I recovered actually three pesticides from the Dal Lake. Right. I have an externally funded project. Now I am trying to extensively sample Dal Lake, and one more lake is called as Anchar Lake here, and we will be formulating the baseline. What is the contamination level of our lakes in Kashmir? Number one. Number two is there is a study actually which was done earlier to my study, that some researchers have detected chlorpyrifos from the indigenous Schizophrenic tiger in Kashmir. Although I was not actually that time, it was beyond the scope or beyond the objectives of my PhD work. I just saw the level of contamination in lakes and in vitro or in vivo effects of pesticides in fishes. But now I have a project. I'm working on that. We have taken up that project that we will see the level of contamination of pesticides in the fishes as well. We will be coming with the data soon. Uh, uh, we have 2 years left for that project, and we're expecting good results out of that. Thank you. Hope to answer the question. Okay, thank you. Thank you. Uh, Thank you, Devendra Saab. Any other question? Kamlesh,
If any question is there, we have. I think we've gone over our time. If there are any, like, we can take one question. If on audio somebody wants to ask, they can ask on microphone. If there are no questions, we could consider the session to be over. On behalf of the participants and organizing committee, I would like to extend our heartfelt gratitude to our esteemed guest who has graced us with such good topic and valuable insight on the topic. And the interaction also with the questions was nice. We express our heartfelt gratitude to you. Thank you for being with us, sir. Thank you.
thank you. Thank you, sir.
Thank you, everyone. The next session is at 12:30. Please join at 12:30. The link will be shared on your email and on WhatsApp group. Thank you, everyone.
Participants, next meeting is postponed. Next session is postponed. Now we have third session at 2:30.